Hello everyone. Today we will discuss about electron transport chain. The term electron transport chain is related to two other terms, biological oxidation and oxidative phosphorylation. Biological oxidation is the synthesis of ATP on the oxidation of foodstuff, in which carbohydrates, proteins, lipids are converted to simpler ones, that is monosaccharides, amino acids and fatty acids. Finally, there will be formation of acetyl-CoA which enters TCA cycle and form reducing equivalents like NADH and FADH2 and then carbon dioxide. Then there will be transfer of electrons from these reducing coenzymes through electron transport chain to oxygen. Here in this process, ATP is generated. That is, from NADH there will be 2.5 ATP, from FADH2 it will give 1.5 ATP. This is known as biological oxidation. In electron transport chain, there will be transport of electrons. Electron transport chain is the complex of mitochondrial proteins which are involved in electron transport. When there is transport of electrons through the electron transport chain, there will be oxidation. When there is a coupling of this oxidation with phosphorylation, phosphorylation is the addition of inorganic phosphate to ADP, there will be formation of ATP. This is known as oxidative phosphorylation. And the complete process of biological oxidation is known as cellular respiration. Let's see some terms in electron transport chain like oxidation, reduction, redox pair and redox potential. Oxidation means loss of electrons and reduction means gain of electrons. Here in this reaction, Fe2 plus ion is converted to Fe3 plus ion by oxidation. And the reversible reaction by gain of electron, it is known as reduction. This Fe3 plus is converted to Fe2 plus. That is oxidation and reduction. The compound which exists in both oxidized and reduced form is known as redox pair. For example, in the reaction NADH plus H plus to NAD, then FMN to FMNH2, here oxidation of NADH plus to NAD plus is coupled with the reduction of FMN to FMNH2. That is known as redox pair. Coming to redox potential, the quantitative measurement of the tendency of a redox pair to lose or gain electrons, which is measured quantitatively. It is known as redox potential or electron transfer potential. For example, redox potential of FAD to FADH2 is minus 0.22, NAD to NADH, that is minus 0.32, then FMN to FMNH2, that is minus 0.30. This is known as negative electron transfer potential. And in the case of cytochrome C, it is plus 0.25. That is known as positive electron transfer potential. This positive means higher affinity for electrons than hydrogen. Negative means lower affinity for electrons than hydrogen. These electrons are transferred from negative electron transfer potential to positive electron transfer potential in electron transfer chain. Let's discuss about electron transport chain. The site of electron transport chain is in the inner mitochondrial membrane. There are four main complexes and fifth one is known as ATP synthase, which is responsible for ATP synthesis. First we will see first complex, that is complex 1. Complex 1 is known as NADH coenzyme Q oxidoreductase. It is also called as NADH dehydrogenase. It has two prosthetic groups, FMN and iron sulfur centers, that is FES. It has 42 different polypeptide chains and it is L-shaped. One arm of this L shape in the inner mitochondrial membrane and other arm in the matrix. So in complex 1, there is transfer of electrons from NADH to coenzyme Q or ubiquinol. Here from NADH plus H plus electrons are transferred to FMN and it is converted to FMNH2. From this FMNH2, then electrons are transferred to FES, that is a protein, then which exists in oxidized and reduced state. From here, it is accepted by coenzyme Q or ubiquinone, which is converted first to semiquinone, that is QH, then to ubiquinol, that is QH2. that is complex 1. Coming to complex 2, complex 2 is known as succinate coenzyme Q oxidoreductase. It is also called as succinate dehydrogenase. It has two prosthetic groups FAD and FES. Here, from succinate, electrons are transferred to FAD. Then FAD, it is converted to FADH2. From FAD to the FES protein, then to coenzyme Q or ubiquinone. This coenzyme Q is reduced to QH2. So, from succinate to FAD, FAD to FES, then to coenzyme Q. Coenzyme Q is reduced to QH2, that is complex 2. Complex 3 is known as coenzyme Q cytochrome C oxidoreductase. It is a multi-subunit protein and it includes cytochrome B, cytochrome C1 and FES. Here, electron transfer from coenzyme Q to cytochrome B through this FES, then to cytochrome C1, then to cytochrome C. From coenzyme Q to FES, then to cytochrome B to cytochrome C1, then to cytochrome C. This is complex 3 coenzyme Q cytochrome C oxidoreductase. Coming to complex 4, complex 4 is known as cytochrome oxidase. It has two hemes, cytochrome A and cytochrome A3. It has also copper, that is CuA and CuB. Here, transfer of electrons from cytochrome C first to cytochrome A, then to cytochrome A3, then to oxygen, that is the final electron acceptor. Then this oxygen is converted to water. From cytochrome C to cytochrome A, A3, then to oxygen, oxygen is converted to water. That is complex 4. So, in electron transport chain, from NADH plus H plus, these electrons are transferred to FMN. FMN is converted to FMNH2. From this to FES, from FES to coenzyme Q, then that is complex 1. In complex 2, from succinate to FAD, FAD is converted to FADH2, then to FES, and from FES to coenzyme Q. Then this from coenzyme Q to complex 3, that is cytochrome B to FES, FES to cytochrome C1, then to cytochrome C. Cytochrome C, from cytochrome C, electrons are transferred to this complex 4, that is first to cytochrome A, then to cytochrome A3. Then to the final electron acceptor oxygen, and this oxygen is converted to water. During the entire process, there will be pumping of protons from matrix to the intermembrane space. In complex 1, that is 4 protons, complex 3, 4 protons, and complex 4, there is pumping of 2 protons. So, total 10 protons are pumped into the intermembrane space. These protons are transferred through ATP synthase, that is complex 5. During this process, ADP is phosphorylated to ATP. So, ATP is generated in this process. So, from NADH, 2.5 ATP and from FADH2, 1.5 ATP are generated. So, this is about electron transport chain. This is today's topic. Thank you for watching.